is really helpful. So we're going to talk about eDNA, the reasons I think that it's really helpful, and the new sectors that I think are utilizing this technology, as well as some limitations to the technology as well. So with that, I'd like to first just say I have no disclosures that I need to make at the moment. So I don't have any financial investment into any of the companies that do eDNA testing. And as I said, I'm a private consultant at this point, so I don't work for any of these commercial labs directly. Okay, so I thought that it'd be more fun to do sort of case-based learning today as opposed to a traditional webinar setup. So first, case one is about conservation. So how we're going to use eDNA in the conservation space. And this really came about when I was early in my career at the University of Guelph, doing research into fish. Basically, how do we tell fish apart? So that the taxonomy of fish, how do I identify those fish? And I was a very stinky undergrad researcher. I always smelled like fish, and I was always pulling scales out of my hair. So it wasn't the most glamorous position, but I really loved my time. But I did end up feeling a sort of bleeding heart scientist moment of watching these fish struggle in the nets and just feeling like there's got to just be a better way. But at the time, there really wasn't. So what we know, right, is that if we can't measure something, it's really hard to improve upon it. So of course, we all remember our Kelvin degrees, but I really like this expression that if you cannot measure it, you cannot improve it. So we need to utilize the ways we had currently to measure the environment. And we needed those ways so that we could measure our interventions. So say, for example, we had a really degraded environment. We see, garbage. We don't see any sort of natural biodiversity. We're not seeing any, cute fishies going around. And we think, gosh, that's really disgusting. Maybe we can put some intervention here to try to rebuild what was lost. So for example, like a kelp forest, we can try to build a kelp forest here. But then we have, of course, the science haters and environmental haters saying, okay, but prove it. Did it do anything, or do you just have a bunch of seaweed now that you've grown? And of course, we know that it's kelp, but yeah, so we'll have these people, right, ask, like, prove it, prove that you did a good thing, right? Did you actually increase the biodiversity, or did you just plant some underwater plants and that really didn't do anything that you said it was going to do? So we need to be able to measure these things. And the previous way we would measure them are things like trawls or electrofishing. So I'm using these water-based examples just because that's the field that I came from originally. So it's just deep-seated in my mind, but we can sort of extrapolate that this is the same in any sector. So if we're talking about reforestation, so we're talking about terrestrial environments, or really any kind of other environment and ecosystem that we could want to restore. So in the water environment, the traditional survey methods that we would use to prove that we have biodiversity uplift or prove that our intervention had some kind of positive or negative or neutral effect is through trawling. So these are these big nets that we use. We put them in the water and we basically capture all of the fish that are there. We pull those out of the water, and then a stinky undergrad or some other specialist like myself would be on the boat, speciating the different species, trying to identify who all is there, at what stage of their development, and then we ideally put them all back in alive into the water body. As you can probably guess, there is a mortality rate associated with this type of survey. And electrofishing is a different method that we use in shallower water systems like streams. And basically it involves a current, two electrodes and a current that passes between them. And it sort of shocks the fish so that you're able to then again capture them, identify them through a taxonomic identification, and then release them back into the environment, with the idea being, again, that the majority of these species would live through this process. But of course, they don't all live through this process, right? And at the time when I was, very young and naive, I thought that this was horrible. I thought this was a very horrible thing, that there were all these fish caught in the nets that would die in the process, and that sometimes we were catching other things in the process and injuring them. So it's a very, I'm sure as you can see, it's a very invasive way to do surveying, to know if the thing you were trying to do to help these creatures was actually helping them. So it feels a little counterintuitive, right? So to me, it was very obvious there needed to be a better way to do this, right? And I was really at the right place because while I was there, the International Barcode of Life project was being built at the University of Guelph, where I was doing my undergrad and doing these research projects on the side. So that was really an effort to try to build up eDNA as a better way of doing these surveys so that we didn't have to be harming these creatures. So let me tell you about eDNA and then tell you how we used it in these projects and how me as the bleeding heart youngin felt better about what we were doing. So eDNA testing is basically it's completely based on the premise that all living creatures leave behind a genetic trace in the environment. So if we take a sample of the environment, we can capture that genetic trace and sequence it to see who was there. So for example, if I just do this, a little bit of my skin cells goes off into my environment. I have a big 50-pound Labradoodle over here who likes to roll around in dead things. So I'm sure that in my environment, there's also, raccoon and skunk and squirrels and all sorts of crazy creatures that he likes to try to bring home with him. Always very fun bath time over here in Toronto. So I have a lot of different creatures that are just, their genetic materials just floating around in my home. And if we were to take a sample of the environment around me, we could pull out that genetic material from an environmental sample and then be able to sequence it and say who was there. So maybe I would find, pardon me, I'm still getting over a bit of a cold, so I'm sorry to be coughing in all of your faces, but thank you for being patient with all of us here in Canada that are basically all sick right now in the wintertime. So I do apologize, but thank you. So we can take these environmental samples, and I'm going to be talking about water samples, but we can take soil samples, air samples, we can take swabs off of leaves, we can take punches of leaves so that we see, genetic hybrids of different plants as they're hybridizing in different ways. So we can take a number of different types of environmental samples, but mainly I'm going to talk about water samples, and that's just because that's the most studied type of sample. So we take some water and we expect to find things that live in the water, so the genetic trace of, say, fish or amphibians, but also mammals go to drink from water systems. And when it rains, DNA that's hovering in the air from birds, as well as from other creatures and insects, will go down with the rain into water systems. So we actually get genetic traces of many other creatures from a water sample. So what we do is we take that water sample, we extract out the DNA through a very simple process called DNA extraction, which is done widely at every genetic lab on the face of the earth. So labs all know how to do DNA extraction very easily. And then from there, we have two questions that we can ask with an environmental DNA sample. The first is a question that is asking, is a specific species of interest present or not present? So it's a black or white answer. And that's really very helpful when we're trying to track invasive species or sensitive species, where we're really interested primarily in one creature. It's a very sensitive test. So we could say, is this invasive eel present in this water body or not present in this water body? So that's one kind of question we can ask from eDNA testing. Another type is asking, who's recently been here? So that type of question is not a black or white answer. That's going to give you a species list. And that species list is going to tell you that within the last week, approximately, which creatures have recently been in that area. Okay. So that's the difference between the two types of testing we can do from an eDNA sample. One is called qPCR. That's the black or white, yes or no question that we're going to ask, is a species present or not present? And then metabarcoding is the other type of testing we can do, and that's asking who has recently been in this area. I'm going to mostly talk about metabarcoding just because we get so much more data from that type of testing, and I think it's cooler. So qPCR is a type of testing that we have been doing for many decades, and honestly, it's not really super cutting edge. It's very reliable and robust, which is why it's wonderful that it exists. But metabarcoding is cool because it's new, so I like talking about that more. So I hope you'll bear with me talking about something that's a little more novel and fun. So so there are some other differences here too, right? So we can just see just sort of logically, we can appreciate that if you're doing a traditional trawl, you're going to need a big net. You're going to need some people that know how to set up a giant trawl. You're going to need a boat. You're going to need some specialists. You're going to need probably a few specialists to be able to take these fish off out of the water, figure out who they are, and put them back in a time frame that allows the majority of them to survive that process. And you're going to need some time to do that, right? Whereas on the flip side, we have something like an eDNA sample, and that's just a water sample that we push through a water filter. So here's an example of a syringe that's filled with water that is the water sample that we would want to know who has been in that water recently. And whoever this is doesn't need to be a scientist. Whoever this is pushing that water through a filter, and that filter is what's going to collect the genetic material that's in the water. Okay, so there are some resources and risk that is different between these two methods. So trawls involve a large vessel usually because there's equipment and personnel that's needed to do the testing. I have here a little mad scientist, meaning like specialists that need to do this stuff. Mad scientists because I think we're all a little crazy if we want to go on boats and try to figure out what fish that tiny little silver fish is. Like I was very bad at this because I could never tell. I could never tell it. It's just a little silver fish. I don't know. So obviously you need better specialists than me to go and do those things. But it takes time. It takes time to figure out what these are. And that can introduce a number of risks. Risks when we're talking both from a financial perspective as well as a personnel perspective. So financially, we're going to need a bigger vessel, more equipment, specialists, and more time at sea time. Okay. And then personnel risk, we're going to need good weather to be able to do this type of survey. Having experts flowing in from different areas introduces risk, as well as, trawls are a very safe method of surveying, but there is always a small risk that something could happen when we're using large pieces of equipment, right? Whereas if we're doing an eDNA survey, we're just taking samples of water from that same area of interest. And we can, if the at-sea conditions are not super great, we can actually just take that water back to the land. So we don't need as much time to be actually on the water. The vessels don't need to be the same size. They can be smaller. They can be, they don't need to be sort of commercial vessels. They can just be, someone in a kayak. And we don't need specialists to do this type of collection. So we have lots of citizen scientists that have collected eDNA samples. Again, it's just water being pushed through a filter. So as long as we do that with sterile technique, so we make sure we're not cross-contaminating our samples, as long as someone understands how to filter in a sterile way and sterilize between filtering events, then anyone can collect a sample. So it makes it easier as well as less risky to do this type of surveying. So right, we can see that for conservation, right, eDNA provides the ability to take a sample in a non-invasive way. So water sample instead of literally taking fish out of their environment to try to make an environment that's better for them. It's a little hard to wrap our heads around now that we know there's a better way, right? So environmental samples are easy to collect. right? They're non-invasive. We don't need specialists. They reduce risk. It's usually a cheaper method of sample collection and data generation. And we can amplify our target species. So we can specifically say we're interested in fish or we're interested in marine mammals. And so we extract that DNA, we can amplify up what we're interested in learning about. And we need to do that because there's just so much genetic material in our environment that we need to sort of be specific about what we're really looking for. And then we match the sequences that we get from that amplification. And from there, for a large portion, we can actually then name that genetic sequence to a species. So we can say, oh, yeah, there was this species of interest that was found, but there were these other 20 species that were found or identified. We then get that list of who's been there. So let's see an actual example of this in practice. So wind farms, offshore wind farms are notoriously difficult to survey. And the data is really helpful because we have commercial fishing industries that historically have been very against these types of developments. So we can just think that a large portion of the fisheries industry may consider offshore wind structures and farms to be disruptive to their business. They sometimes can get in the way of specific fishing routes that may be of interest. They also, maybe disrupt the fish's migration or, spawning habitats, although of course that's taken into account when a lease area is generated. So there has been sort of this, I'm going to say it really nicely, but everyone can read between the lines. There's been some low levels of tension between wind farm developers and the commercial fishing industry. And I think that that's changing. That narrative is changing, and eDNA is a large part of why this relationship is getting better over time. So one study that was done at the Blyth Offshore Demonstrator in the UK, that's a research. So there's a large offshore wind farm, and they've designated part of it to research. So the demonstrator is where research is done. And there was this one study that looked at trawl. So it's traditional trawl-based survey method for measuring biodiversity in fish. Versus eDNA, so collecting water samples for eDNA and seeing how the results of those two types of surveys compared. So one of the just outright differences between these two survey techniques is that trawls can't go into the turbine array, pardon me. So these structures are too big and there's too much equipment under the water, and the trawls could disrupt that or get caught in different parts of the turbine array. So trawls actually just can't get into the turbine array to measure the biodiversity and what's happening within the array. Okay, so right off the bat, a water sample can do that. So eDNA already has an advantage, just that it's able to see what's happening under the surface in that area, that specific area. It was also shown that trawls required more time and personnel. Again, that's just logical, right? You got to set up a big trawl, you got to set up a big net, you need specialists, you need time at sea. So that just makes sense, right? It just makes sense that eDNA would just be a faster method. But it was interesting to see that there were more species that were detected with eDNA, and that's really the important thing. So what the study showed is that we have here on the left side of the Venn diagram the eDNA species that were detected. So overall, there were 54 eDNA species or species that were identified with eDNA that were detected. And then the trawl was able to detect 26 species. There was an overlap. Of course, that makes sense too, right? They're both good methods of detecting the biodiversity. But eDNA was able to detect 35 more species in addition to the trawl, whereas trawl was able to detect 7 species that eDNA was not able to find. So there are some differences there, one being that species that are small can escape the trawl quite easily. So in this 35 that the eDNA picked up, a lot of them are smaller fish that actually just can swim through the trawl nets. So that makes sense. And then the benthic species were also picked up with more abundance. So bottom-dwelling species were able to be detected easier with eDNA as opposed to the trawl method. The trawl method was, on the other hand, the trawl method was able to detect species like sharks and rays more easily than eDNA. And that's because sharks and rays they actually shed less genetic material in their environment. So not all creatures are able to be seen using eDNA because they just leave less DNA in the environment as they pass through it. So sharks and rays are part of those groups. Yeah. So pros and cons to each method, but it was really helpful to demonstrate that eDNA is able to pretty reliably detect more species than a trawl, as well as get into the turbine array. And within the turbine array, a very interesting and compelling argument that supports these types of structures. It's not going to blow anyone's mind, but basically these structures create an artificial reef system. And that's actually really good for as creating a sort of nursery environment for a lot of fish species. And when we pulled out what those specific species were that were using this environment as an artificial nursery, we found that a lot of them were of commercial fishing industry, like were very valuable to the commercial fishing industry. So what was actually happening is that these structures that the commercial fishing industry had been really against, these structures were actually creating a little nursery for the fish that industry is later going to want to harvest. So being able to generate that data specifically within the turbine array and present it back to the stakeholders in the community that historically haven't been real big supporters of these types of projects, it did allow for the conversation to begin changing to being more supportive of these projects overall. Yeah, so that was a really great outcome from this research. And this research was, it was done a few years ago, so it continues to be built upon, which is wonderful to see. Okay, so let's move on. Okay, let's talk about some limitations of eDNA. And we're going to talk about it in the environment of the desert. So I used to live in Arizona and I love the desert. I'm a just terrible Canadian. I miss the sun and the heat. I like when I feel like I'm walking out of my home directly into an oven. So Arizona was definitely the right place for me. Um, it's currently a snowy, icy hellscape here. So I'm in the exact opposite. Yeah, I'm in exactly the wrong place than I want to be. But I thought I'd bring, I do like to try to bring the desert with me as I give talks because I loved it so much. So I'm going to give one of the limitations of eDNA in the desert. So, okay. So the Alameda whipsnake is a threatened snake species. It's mainly found in California. Alameda makes sense. Okay, so when we're thinking about threatened species, eDNA can be a really good way of detecting those species because we don't see so many of these creatures in the environment. That's why they're threatened. Like their numbers are literally less. So when we're thinking about population density, we're talking really low densities, which means that if we're setting up camera traps or if we're sending out specialists to try to track down these species of interest, those can be pretty hard ways to find these creatures just because there are so few of these creatures. So how do we track snakes with eDNA? Well, we can't use water samples, especially if we're talking about the desert. There is water in the desert. Please don't get me wrong. I'm not, I'm not saying that there's no water. I'm just saying that there is less of it. So it's a less reliable method of environmental surveying for eDNA. So what do we have? We've got some kind of rocky sand. We've got air. Those are kind of the two major environmental sample types that we can use for eDNA analysis. But spoiler alert, those are not great ways of getting eDNA testing done. And that's really because the desert is quite a nightmare for eDNA. as well as shedding reptiles. So shedding reptiles leave large amounts of genetic material behind as they shed, but then as they like once they're past their shed period, they don't leave that big a trace. So and they leave then at like a clump of genetic material in one place. So it can make it artificially look like there is one of these snakes somewhere when really they've moved on. They may be many weeks away from that area. So we've got some caveats. So is eDNA really a good way for us to understand if the species is there? And the answer is no. So spoiler alert, Pobody's nerfed. I just rewatched The Good Place. I don't know if anyone has watched this show, but I love it. And I love that expression now. So Pobody's nerfed. This testing, eDNA testing is not perfect. Um, DNA is degraded in UV. It's why we as humans can get skin cancer from sun exposure. So one of the reasons I love the desert is that there's a lot of sunshine. So that is a real nightmare when it comes to trying to do eDNA work, right? Because the DNA is being degraded really quickly. Also, some species just are not good shedders. So we talked about sharks and rays. We talked about shedding reptiles. There are other species too that just because for one reason or another, they don't shed as much DNA or the DNA that's shed is degraded so quickly that they just are invisible to eDNA surveys. Okay, so not all creatures are really going to be able to be detected with eDNA testing. Yeah. And then there are substances in the soil, water, and excrement that can degrade or inhibit eDNA work. So things like bats, we may try to collect their samples in the environment by soil sampling, for example, or sampling right at a cave mouth when they're excreting. But within their excrement, there's actually an inhibitor, so we're not able to do that testing very reliably for bats. So we just would recommend other tools if that's the case. So if we're specifically looking for the Alameda whip snake, I'm going to say, cool. I hope that you are able to find those snakes, but eDNA is not going to be the way you're going to do it. Okay. But that's not to say that for every threatened species, eDNA is not a good mechanism of detection. So this is a Blandings turtle. I think they're quite cute. I think they're quite cute because they're also in Canada. And in Canada and a lot of our lakes, we have snapping turtles, and they are sort of cute, but also they can apparently get big enough to like bite your fingers off. And as a child growing up in these lakes, I was always terrified then of turtles in general. Um, childhood environmental trauma, I guess. I don't know what to call it. It's crazy. But these are very cute and they don't bite. And so I love them. And they've got these cute yellow chins. And I just think they're adorable. But unfortunately, they're also endangered. So that means that they're really hard to find. And actually, they do such a good job. I mean, this one is doing kind of a terrible job, but in general, they do a really good job of blending in. So they really do got those camo skills. They just like blend in. They're really hard to find. They're endangered, so their numbers are really small, and they can hold their breath for a really long time, which means that they can be really, really, really hard to detect. So as land is slated for development here in Canada, one of the things that needs to be checked upon if there is freshwater lakes and ponds is the presence or absence of the Blandings turtle. And developers hate this because understandably, it's quite a nightmare to try to find the Blandings turtle. And it's always challenging, of course, to prove a negative. There is no Blandings turtle here. So that's impossible. So we do the best that we can. And really, the best that we can is by using eDNA. So eDNA has been now charged as the way that we're going to be looking for Blandings turtle in Canada, specifically in Ontario at the moment, but it's being rolled out across the country. And that's because it just works better than traditional methods. So we can take samples of water bodies around an area that's slated for development, and it's a more reliable method for finding is there genetic presence of a Blandings turtle, even if we do not visually see the Blanding's turtle there. And this was really, this was really based on work done by Robert Hanner at the University of Guelph back in 2021, who showed basically that, yeah, eDNA was just a better way of doing these surveys specifically for Blanding's turtle. So it's not to say that eDNA is not good for invasive species or sensitive species, endangered, threatened. It's just to say that there are limitations of the technology. So it's not going to be good for every species that is of interest for environmental surveys. Yeah. Okay, so let's get into how we're using this data, because we know that this type of testing, eDNA testing, can generate these species lists. So we get a lot more data than a traditional method like a trawl. Trawl, we're only getting information about fish that can be captured in a net, but eDNA is looking at any living creature that was in that water body or close to that water body. So we can look at marine mammals, we can look at invertebrates, we can definitely look at fish and other vertebrates, but we can look at a large number of other things. So we get a lot of data from this type of testing. And it's been really wonderful to see the scientific community come together to create publicly available databases to house this data so that it can be used across different research institutions and by anybody. So we have open access to a lot of these different, databases. The primary one that the two that I've been using mostly are BOLD and GBIF. So I'm going to give you some examples of how the data has been used, but it's really nice to see that there's a number of different places where data is housed. And that does lead to the possibility of people accessing data and coming up with machine learning algorithms, for example, that are based on really large data sets, as well as generating new insights that may be missed by the researcher's primary goals for that data collection, for example. So let's talk about one of them. So this was work out of the BOLD and GBIF databases. And basically the question was, will eDNA work well at different depths at the same location? So eDNA is still a novel tool. So it's really only like maybe 10 to 20 years old at this point. And of course, 20 years ago, we're just talking about just barcoding species. So we're just talking about figuring out unique genetic sequences of certain species. When it really started becoming more widely used was about a decade ago. So it's still a relatively new technology. And that means that we're still learning a lot about it. So one of the questions, rightfully so, is how far does eDNA travel in a water system? So if I'm taking a sample at the surface level, but really I'm interested in benthic species, so species at the bottom of the water column. If I take that sample at the top, am I going to get that information? Am I going to learn anything about the composition of the biodiversity at the benthic level? And these are things that are being asked and being thought about. If there's currents, how far does the DNA left behind by creatures, how far does it travel? If I take a water sample 5 kilometers away from a marine protected area, am I going to pick up all of those species from the marine protected area and then maybe artificially think that there's a very rich biodiversity area when really it's not? It's a degraded area that I'm looking at a restoration project. So these are questions that are being asked in the field and being sorted out with the use of these large databases, open source databases. So a lot of grant-funded research will funnel back in their data into these databases so that everybody can access it. So this was tackling one of the questions of, if I take a sample from the surface, will it give me information about the bottom? And the answer is no, which is good. It actually, what they did is they took 10 different sites around Monterey in California, and they took samples at varying levels of depths and they compared the community composition. So community composition is just the species that were found and how they relate to each other. And basically it showed that there were statistically different community compositions depending on depth. So they took samples at surface level, they took second samples at either 20 or 40 meters, and then they went to a shelf area and they took at the shelf, which was around 200 meters, and below the shelf more than, they just said more than 200 meters of depth. So they were able to show that the DNA compositions at those different depths was statistically different, meaning if you're interested in benthic species, you got to take a benthic sample. And if you're interested in surface species, you got to take a surface sample. And good and bad, right? Good because it tells us that we're not getting cross mixture there. So if we are really specifically looking for the community composition of a benthic habitat, then if we take that sample there, we're not going to get contamination from any species that are surface dwellers. And vice versa. But bad because then, for people that are actually just interested in getting sort of a whole water column understanding of a community, they can't do that with one sample. But there's a way around that. You just take samples with Niskin bottles at different depths and combine them and put all of that water through one filter, and then you get the answer. So different ways around that, but it, it's just showing that the data is helping to drive our insights into how this technology can be useful, the limitations of the technology, and how we can get around some of those limitations. And I really like this example. It's not eDNA in particular, but there's interest in adding eDNA into these types of analyses. So this was a data set from GBIF that was used, and it was looking at nature-based tourism in Costa Rica, and it was using social media pictures from people posting around Costa Rica, and they were posting pictures of birds, and I love that. Especially because the birds in Costa Rica are just so amazing. So I think that that's very cool. And what they were doing was they were matching those pictures to databases that would use AI algorithms to name the species that was within those photos, and then using these pictures as a proxy for biodiversity. And basically what it showed, unsurprisingly, and of course there is a bias that people going to Costa Rica may be more interested in biodiversity as part of their tourism, but then, unsurprisingly, right, what this showed is that the more biodiverse areas were visited more frequently by tourists and were posted more. And there is some interest in trying to monetize that in some way. So trying to look at green investment. And green investment is about funneling money into environmental projects. So projects that have a positive environmental impact, but also will have financial returns. And eDNA is one way that's being leveraged currently to help ensure that financial investors are making returns. So let me go into one of those examples in specific. It's a little out there, but it is how I see people getting rich from environmental-based projects. So I think that the future of the financial market is environmental based, and this is one example of how it can work. So the World Bank has come up with something called an outcome bond. An outcome bond is a bond that financial investors can invest in. They get a guaranteed return back. That's just a normal bond. But they only get that return if the outcome is met. If that outcome is not met, then they get a minimum return. So anything extra is based on the outcome of some project. The project in question for this outcome bond was an Amazon reforestation project. So there was a group that was doing reforestation in the Amazon. And anytime we talk about reforestation, the way to monetize that is through the carbon market. So through carbon credits. And actually, there's a big company that needed some of these carbon credits. That was Microsoft. So Microsoft said, okay, great. dear Amazon reforestation project people, we're going to buy your carbon credits and we're going to buy them at this rate. And we are going to use some of them, but there will be extra. And if there are extra, based on your calculations of what you're going to be doing, we will give those back to the World Bank and to the financial investors who have invested in this bond. And the Amazon Reforestation Group was like, awesome. Okay, well, that sounds really wonderful, but we need some startup money. Like we can't just start. We need to actually have money to start this project. So it's great that you're going to buy the carbon credits at the end and like we'll make some money, you'll make some money, investors will make some money. Like this is all good, but we need to start and we can't start without financing. And that's where the World Bank came in. So they said, okay, great. We'll fund your startup costs. You need to pay us back for that though, but we'll fund it to start up and we'll guarantee financial investors their money back plus a little extra. And dear Microsoft, if there's any extra that comes out of the carbon crediting, you'll give that to the investors. And in that process, in that cyclic process, it has so I think it's in year 3 at the moment, and it has so far been as successful as was anticipated. So it hasn't been more than they expected, and it hasn't been less, and that's really good. And part of this process, there was a huge amount of due diligence needed to make this work. Lots of parties involved and lots of money involved. So there was over $225 million involved. So lots of money involved, lots of large organizations involved, and so lots of due diligence. And part of the due diligence was making sure that the Amazon reforestation efforts weren't just going to plant some trees and then things were going to not do what they said they were going to do. So we've seen some of this happen. There were projects in the Philippines, for example, where they, planted, hundreds, thousands of these mangrove trees, baby mangrove trees, all of which were washed away in 1 year, and maybe only 2% of those trees actually took hold. So we know that there are projects that just have not been successful. So as part of the due diligence, Microsoft needed some other way of monitoring the success of the reforestation other than simply number of trees planted. So eDNA was a way that they suggested to do that. So basically, soil samples were taken over time, and every 6 months, soil samples are taken from the same area and from new areas that are being regenerated, and they're looking at the soil microbiome to see an increase of biodiversity uplift, as well as a decrease in pathogens that are known to be within those soils. And that's another indicator that we're seeing this restoration of this environment. Okay. And this is a really important example because, again, like I said, I think that the future of the carbon market is going to be a nature-based credit market. So there's already talk about biodiversity crediting, but I imagine a future where people get quite rich from doing restoration projects. I like to think, clearly, I'm an optimist over here, okay, but I like to think that that's possible. So the majority of why I think that's possible is because the financial markets are hugely based on nature. So, where would Starbucks be if all of their farms go under because of climate change, right? A huge amount of our financial market is nature-based. And the World Economic Forum is the leaders of the stats that help us really cement that way of thinking. 50% of the global economy is under threat from biodiversity loss. So that's pretty huge, but it's not all doom and gloom, right? They come up with strategies to try to help the market be resilient to that. So they've come up with, 5 sort of transitions or recommendations to help the financial market and help our supply chains sort of de-risk from an environmental and biodiversity perspective. And if those are undertaken, if those 5 strategies are undertaken, then the anticipated revenue is about $3 trillion, as well as several hundred million jobs that would be created. So So it's not just me saying I think that our economy is very nature-based. The numbers say that too. And if we adopt a nature-based solutions, we actually build not only upon economic growth, but also jobs as well. And that again just makes sense, right? If 50% of our supply chains are nature-based, then if we invest in those things, we would expect huge returns. So again, I think that eDNA plays quite a role in a lot of this. And yeah, and so I think it's a really very cool new tool. It's innovative. It's really only been commercially available for about a decade, if maybe commercially sort of a little less than that. So it's a newer tool in the marketplace. It's non-invasive, so we don't have to hurt the little fishies as we are, monitoring them. It's also easy. It's easy to collect a sample. So I myself have taught school children how to do it. So it's very easy to do. Anyone can take an eDNA sample. It's a safer and cheaper method for environmental surveying in general. Not always, but in general. And it's not the best tool for every project. So we did talk about some of the limitations of this technology. So depending on what people are trying to evaluate, this may or may not be a good tool for that specific project. But in general, it's a good tool to consider. Yeah. And then it generates lots of data. So we all love lots of data. It's generating huge amounts of data, lots of which is open sourced. And so it does create the possibility for new algorithms to be developed and new ways to monitor our environment and understand our world, the world's biodiversity in these new ways because it's so easy to access this data at the moment. And there is a huge push for offshore wind developers to be submitting their data into public databases. Historically, that hasn't been the case, but starting in 2025, any new lease areas that are being developed in the US will have to publicly submit their data into these databases. So you can imagine they have huge amounts of data for their projects. And then it's a really easy way of measuring biodiversity. And as we look at nature being more and more involved in the financial market, they need things to be accessible. These are mainly investment bankers that are pretty, understandably, this is not their field of expertise. So they need easy ways to access environmental data that they can understand. And I think that eDNA is a tool that allows the financial markets to access nature and understand it and use technology and tools to help make sure that they're getting good investments and that the projects that they're investing in are doing what they should be doing. Yeah. So so that's what I had over here for the cool things that I'm seeing in the eDNA world. But I'm very happy to take any questions that you might have at the moment.
Awesome. Thank you, Rachel. That was great. Yeah, any questions?
I'm just curious. I listened to Radio Lab, the radio show, and their most recent episode was about this sort of field of aerobiology that kind of dissolved when the founder of it got in a plane crash and disappeared forever. Um, but like one of the things that they wanted to do was to have like a sort of like disease forecasting tool, like we have a weather forecasting tool. And so I'm curious if eDNA is something that you can do from air samples and like how what the potential is to like automate that. Like, could we put an eDNA sensor on a flux tower, for example?
Yeah, that's an awesome question. It is being worked up right now. So currently air eDNA samples are a thing. They're mostly used they're all used currently in the research setting. My understanding, though, is that there is a large organization, commercial eDNA testing lab, that we'll likely release an air eDNA product this year. So what it looks like, we have two different ways that we can do that. One is passive sampling and one is active sampling. So passive sampling is something like at a cave mouth, having some sort of collection method, something that would collect air as it moves from inside to outside of the cave. So just taking advantage of currents that are already going to be happening, and then looking at specifically bat DNA that's floating in the air and would be captured passively by setting up a collection method at that cave mouth and having it there for, say, 3 days at a time. Cave mouths are usually pretty dark, so we don't need to worry about DNA damage. They're usually pretty cool, so we don't need to worry about the heat damage to DNA. So that's a really good method. Active sampling would be something like having a fan push air through a filter or having a filter method be moving through the air. So for example, I was working with a group that had drones because drones are cool and they had them. And I thought that'd be fun to try to work with them on something. Because I don't know anything about drones and I wanted to learn about drones. So so we set up a filter on the bottom of a drone and sort of drove it over the surface of a desert area. Because again, your girl really likes the desert. And what we found is that that was not a very good way of collecting DNA in the air. So wherever the Journal of Negative Results lives, that was research to go into there. So maybe that form of active sampling was not good, but there are other active sampling methods that are good. So for example, we know that there's one group that's putting active sampling air DNA methods, sample collections on the top of windmills. And this is looking for birds. So they're trying to figure out bird strikes versus birds that are escaping the windmill. And sorry, they can see strikes because we have carcasses then at the bottom of these windmills, but we're not sure exactly who's managing to escape these strikes. So having a fan and a filter set up at the top of a windmill and being able to have that being collected and protected in a certain way so that we are maintaining the DNA that's collected through that sample, and then see if that could be a good way of getting data about the different species that are moving around a windmill. So there are definitely products being developed. I imagine that 2025 is a year where we're going to see some of that hit the market. And they're going to be in fields that need a solution. So I think that wind energy it's just an area where they need a solution. And bats have just historically been very hard to find using eDNA. So having a method that can do that, whereas again, the current method is you set up a giant net in front of a cave and then you capture all these cute little bats and handle them. And you can imagine that there's, quite a large risk involved with all of this. As well as like just feels kind of brutal and a little bit antiquated to be capturing these animals that you're trying to monitor and protect. So they need a better way. So I think that we'll see these products come out specifically for those use cases. It's a great question. And I think that it's coming. Yeah. So stay tuned. I think that that's going to hit the market this year.
Thanks.
Thank you.
I have a question. I thought the results you showed about the DNA being different at depths was really cool. But with depth, there's all kinds of things, including stratification, that could be exacerbating that difference. What do you know about the difference between, you had mentioned this, but about like how far, like at the surface layer DNA is drifting, and how much bycatch are you getting from, like, your example of like there's a preserved area over here and you're sampling this area, and how much of that bycatch are you getting?
Yeah, so that is something that is currently being worked on. At the moment, the kind of going way we talk about it is that within 5 kilometers, you can't reliably know where your eDNA came from. But once you're past 5 kilometers, you can assume that if you're detecting it, it's likely from within a 5 kilometer radius. Pardon me. So of course, there's a number of factors that go into that, right? I mean, if the water is very warm, that is going to be, DNA is going to travel or survive longer in that environment and then potentially travel farther than if the water is really cold. Similarly, sunshine, right? We know that UV degrades DNA. So if it's a really sunny day that you're doing your collection, the DNA you're collecting is going to be from a radius that's smaller than on a day or a week that's been really cloudy. So there's just so many factors that it can be really hard to have one answer to that question. It's really quite situationally based. That is though why on the back end, we use a metric called relative abundancy. So what that says is of the DNA that was collected from this sample, how much came from each of the species that were detected. And that gives us an idea. So it's not a direct correlation, but it gives us an idea of how maybe far or close those species were. So for example, if I'm doing a study and I am trying to gather data to help the commercial fishing industry get on board with a new wind farm development. And I really want to look at tuna. I want to look at big tuna that they're going to be catching, the big guys. And I know that they're big, so I know that they're going to shed more DNA than the little, I don't know, some other little silverfish that I know is also quite abundant there. If I take a sample and I see that there are low levels of DNA numbers coming from tuna relative to these tiny silverfish that I know are kind of everywhere, and I know that they're in the area. I saw them as I was taking a sample. And what that tells me is that likely that tuna was farther away. And that's just a relative frequency of the DNA amounts. So a proportion of the DNA that's coming from those two species. Again, you can see why this is not a metric that's used like hard and fast in any way, because I'm using a lot of very soft language as I describe this. Yeah, it's just ways that we can help interpret the data for researchers and for the data users. But in general, we're talking pretty regional information. So, if we're doing research on a marine protected area versus an area that hasn't been restored or protected, if those two areas are 5 kilometers apart, I'm going to say maybe eDNA is not the right tool to use for this type of study. Okay. But if they're 20 kilometers apart, I'm going to say with confidence that is probably totally okay to use this tool to help evaluate those two places. So again, just good, we all know, just good survey design, just good experimental design is going to help sort of drive down any of the biases that we can see coming from things like, the movement of DNA in the water, for example. Yeah. It's a good question, though. I get it all the time, and I hate giving the answer because it's like really wishy-washy. Yeah, yeah.
Awesome. Thank you, Rachel. Looks like we're at time, but we really appreciate you speaking to us today.
Oh, thank you so much for taking the time with me today to dive into something I think is so cool. I really appreciate you all. Thank you.
Thank you. All right. Bye.
Bye. Bye.